to people that have been here before, it's good to see you again, but there are a lot of faces that are unfamiliar to me, so I hope we can all gain something out of what will be a repeat talk for some and new for others. My background isn't actually as an academic, despite the title. I was born, ignore the West Middlesex accent, I was born in West London, but very young, moved down to Devon. And my teenage years were spent in misyouth, a bit like Tullis's, point to pointing down in Devon. So that's where I got my love of horses. But in more recent years, so certainly for the last 30, I've been based with my partner Jill up on our small holding in Kenmore, Cumbria, where we have been breeding bay horses. My background actually professionally is as a biology teacher, and with my shared interest in biology and horse breeding, when we got involved in Cleveland Bays, we asked ourselves the question, well, actually, what do you do with these really, really rare animals? And it was that question, which I'm sure a lot of you have had going on in your minds, that led me to dig deeper and led to unraveling a lot of the story that I'm going to tell you today. So things that we're going to focus on today, we're all in love with our breeds, but specifically to us, we're managing breeds in closed populations. We've got pure stud books, there's a ring fence around it, and with very few exceptions, nothing gets inside that ring fence from outside, because if it's coming from outside, it's carrying outside non-purebred genetics. And that comes with problems. I want to talk about how we can monitor those problems and also come up with strategies to deal with it. And then look forward and project forward into the future so that our breeds are as sustainable as they possibly can be, given how compromised they are. And some of them are very compromised. I apologise now for pictures. 90% of them are gifted, 10% are stolen. If I've stolen them from you, please forgive me, okay? Small populations. One of the really inherent problems of a small population versus a big population is that they're going to lose genetic diversity much more quickly than the big populations. And that's through a biological process called genetic drift. And that might be familiar to some of you and a complete anathema to others. And what I'm going to do in a moment is explain as simply as I can genetic drift. But it happens all the time. And genetic drift will lead to a loss of genetic variation, and that in turn leads to inbreeding and to these traits, these deleterious traits that raise their heads, these genetic diseases in inverted commas that we do not want. So these deleterious traits actually lead to a loss of fitness, a lack of fertility, higher foal morbidity, and there is an increasing body of scientific evidence that shows that. And I know this morning Lucy and Tullis have both referred to recent research in the thoroughbred world that is just as applicable to our native equine breeds. So, genetic diversity. I like Tullis's DNA model here. And if we strip that down, that double helix, for the horse, there are 2.3 billion, not million, 2.3 billion genes. They completely genotype the horse now, and they know where that is. But if you stripped all those genes out, if we imagine them as coloured beads or coloured Smarties and stick them in a big heap from any one individual horse, you're going to have different frequencies of different colours. So in this example, if we go down the Smarties analogy, we've got an awful lot more red Smarties in the heap than we have the purple and yellow Smarties, and there are even less green Smarties and blue Smarties. And out of all of those, if we breed from that pool of genetics, we're most at risk of losing the blue Smarties and the green Smarties that we've got the least of in the first place. If you look at the double helix, there are two different positions where it joins up on the ladder. We carry two what we call alleles.
Sorry, do you mind speaking in Denmark?
Yes, I can turn the microphone on and stand in front of the mic if you can't hear me, not a problem. If we look at a mare and a stallion, and I'll go with the stereotypes of blue for a boy and pink for a girl, okay? So our mare, at any one of those positions on the DNA, is carrying allele A and allele B. Whereas our stallion is carrying C and D. So up in that top, the top row, completely unrelated animals. So if we just imagine there is no relatedness whatsoever, no common founders, they do not share common genetics. So we've got A, B, and C and D. And if we cross them, we're going to do two matings. Let's do the first mating. If we look at the foal that was produced in the first mating, A came down from the mare, and D came down from the stallion, and at that single position out of the 2.3 billion, it's A and D. Okay, now breeding season goes by. We cross those same two parent animals again. Obviously, they carry the same genetics at that position. That isn't going to change. This time, the mare has passed on B, and the stallion has passed on D. And if you do a real quick add up, we've still got 4 genes, 4 alleles in the bottom row, but we've got 1 copy of A, 2 copies of D, and 1 copy of B. C has disappeared. It's disappeared completely. And obviously, it's not there to be passed on into the next generation by these animals as they become parents in their own right. So that's in the raw genetic drift. If you take the Smarties analogy, but think of marbles instead, if you start off with a bag containing 10 marbles and you breed from those marbles, right? We do several generations of crossing. You can actually see that halfway down, we've now got 10 marbles still in the bag, but we've only got 6 different colours. And if we carry on more generations, eventually we've ended up with a situation where we've got a lot of red marbles, a single blue marble, and the rest of the colours have gone. Now, if you think back to that original heap of Smarties, we had a lot of red ones. And that's what's happened here. We've actually increased the frequency of the more common alleles, and we've lost the rare ones. So the rarer ones are the ones most at risk of being lost through genetic drift. And that leads to reduced adaptability, term it loosely as environmental change, but all sorts of changes. We lose disease resistance and the ability to adapt to diseases as it comes along. We lose the ability to have breed for different circumstances, and we get inbreeding depression as inbreeding accumulates within a breed. And we know that increased inbreeding leads to a reduction in fitness, increases infertility. It makes it harder and harder to get their mares in foal. But even if we are getting them in foal, it's even harder for these mares to carry the foal full term. And then postnatal, actually the foals to then go on into maturity to breed themselves. Some folks were talking this morning about how important it is to parentage test through DNA. And I agree with you wholeheartedly. There are a lot of breed societies that collect DNA samples, be that blood or hair, and they send them off and get them banked, but they don't actually get them analysed. And with the breed societies that I work with now, and there are many, what we're encouraging them to do is to retrospectively actually take the profile. But in terms of profiling, parentage, if you think of that figure of 2.3 billion genes that I talked about before, we're actually only testing 16 microsatellite markers, so it is a tiny fraction. And actually, for some of the things that people were alluding to this morning, the type of DNA parentage testing that's going on routinely at the moment is great for parentage testing, but it's inadequate and you're not able to detect these genetic deleterious alleles necessarily just from simple microsatellite parentage testing. This is a genuine Cleveland Bay profile. I spent many years on Cleveland Bay Council and Breed Committee, and this was from the Animal Health Trust when it was in being. Old dot matrix printer, this is absolutely genuine, but I purposely anonymised it. But I've also tabulated it. This is the same profile and slightly easier to understand. So down the marker column, we've got the panel of 16 markers, all got funny names, VHL20, HTG, AHT4, for example, was an allele that they discovered at the Animal Health Trust. So they're just named from how they were discovered. And in that second column, the sire column, those are the two alleles that the sire is carrying for that particular marker. In the middle is the progeny of the sire and the dam that's on the right. So let's just work, we won't go right through it in mega detail, we'll just go through a few. Let's start at the top. Up here at VHL20, the sire is carrying I and M, the dam is carrying I and P. And they'll only pass on one of the two that they're carrying. So what's happened? Well, we've got a P there in the progeny. That must have come from the dam. And there's an M there, that must have come from the sire. They couldn't have come the other way around because the other parent wasn't carrying those two alleles. Go down the next one, HTG4. The parents actually both carrying the same two markers, K and M. But they were both passed on M. So that the foal at that position has two copies of the same allele. That's what we call being homozygous. And one of the signs of inbreeding is that you become increasingly homozygous. And if you work down through that, checking each of these markers off, you can actually see that it's a real profile. The foal actually could be, and probably is, the progeny of these two animals by checking it out against 16 markers. So that's one that works. That's what when it comes back parentage verified, that's what it means. Okay, another real example, this time it's parentage excluded. Start at the top again. So the sire's carrying I and N, and the foal's got an N, so that's a possibility. But hang on a minute, the dam is homozygous for N, so could have only passed them on. Where the heck did P come from? Certainly didn't come from either of these two parents. What about at the next marker? Well, the sire was homozygous for M, but the dam was not. They carried K and L. So KM is quite possible. That could work. But if you carry on down there, oh, wait a minute, what's going on here? The foal's homozygous for J. Both parents weren't carrying J, were they? Because the dam was homozygous for H. So if you work your way down through that list there, there's a lot that doesn't work. That's excluded. That's what happens if it goes, it gets sent back and you know, what jumped the fence? Right. This is the one that I wanted to build up to show you because this is very real. It is Cleveland Bay. All of the animals on here are dead and long gone, but it's a classic example of what happens when you've got a highly inbred animal. Same thing, I've just tabulated it in the same way. So, VHL20, and the parentage works out on this, by the way. We don't need to worry about that. But I want you to look at how homozygous the parents are, and then how homozygous the progeny turns out. Because the sire, let's do the dam first. If you quickly count down, I made it 3 homozygous markers, MM. Oh, if there's a question mark in it, it's because it's so early, not all of the markers were tested for. But we're homozygous here at J, with J's, we've got K there, and some of the others aren't tested. But the dam is homozygous for 3 positions, 3 of those markers. Quickly look at the sire. I'm pretty sure the sire is homozygous at 6, NN, MM, GG, II, IL, and LL there. So dam's homozygous at 3, sire's homozygous at 6. Now count the foal. Because I make it 12 out of 16. So we've got 2 parents that aren't desperately inbred. Sire's more homozygous than the dam, but we've got really homozygous progeny because of the inbreeding. And if we go back to the bag of marbles analogy, in that particular case, we've got two animals at 16 markers, each animal carrying 2. So there's 64 marbles in this particular bag, okay? If you actually separated them out into the colours, the different alphabetical letters in the bag, there were 35. The sire brought 32 different letters to the party. Brought 32 markers of 26 different colours. The dam brought her 32, and there were 25 different colours. So there was still quite a lot of variation there. But in the foal still got 32 marbles, but we've now only got 20 colours out of the original 26 plus 35 that we brought to the party. So we've lost a lot, and that's what actually happens when we become increasingly homozygous. The rare colours are gone. So that's a result of inbreeding, but what do we actually mean by inbreeding? And in fact, all of our animals are inbred, because that's how our forebears created our breeds, probably by a process that's better described as line breeding, to fix time. Okay? But an individual is inbred if its mother and its father share a common ancestor. So in other words, if I look at my pedigree, were my mum and dad related somewhere way back at the time of the Norman Conquest? We can measure it, we can quantify it. There was a biologist called Sewell Wright who came up with the inbreeding coefficient, the concept of inbreeding coefficient in 1922. He was an American biologist. And yes, we can get an inbreeding coefficient, so we can quantify it, but that's what it's trying to quantify. It's just how much common genetics is there on the mum's side of the pedigree and on the dad's side of the pedigree. Right, so let's go back to our original model. We've got two completely unrelated animals in that top row. If we cross them, the next generation shouldn't be inbred because the parents weren't related, so they shouldn't be carrying common alleles. But if we cross that next generation, so in other words, if we made filly number 3 and colt 4, then that offspring, 5 and 6, they're going to be inbred because the two parents are related, they share common parentage. And as we said before, we've got this big reduction in genetic diversity. We saw that at DNA level, reduction in breeding fitness, and it's going to greatly increase the problems of deleterious alleles being expressed. You saw a different version of this slide this morning, the extinction vortex. It's that downhill spiral. So if you start off with a closed population that's already compromised, already inbred, if we increase or continue inbreeding within that breed, through genetic drift, we're going to lose genetic diversity. There's going to be a reduction in fitness, reduction in population adaptability. We're going to get a lower reproductive rate, higher mortality. We go on down the spiral. And I see that spiral every morning. I'm looking down the plug hole of the sink when I have a shave. That's where the breed's going to go eventually, if we don't do something about it. Deleterious alleles, what the heck do we mean by those? Not all genetics are inherited in what I call a Mendelian way, not all deleterious traits are. But if we take something that's relatively simple to understand genetically, like for example, to put it in the real world, with the Fells and Dells, for example, lethal foal syndrome, that we now know much more about. Okay? That's inherited simply Mendelian. And that, for example, if we were thinking about it, is we've got the mare that's carrying A and B, but instead of carrying C and D, our sire is now carrying C and X, and X is, for example, the gene for lethal foal syndrome. Okay, let's do the cross. In that first generation, the mare's passed on A, and the stallion's passed on X. So we've got progeny here, female progeny that's AX. And in the next season, we cross the same two animals. Mare's passed on B, and the stallion's passed on X. So both of the progeny are carriers But because there's only one copy of that damaging deleterious allele in either of the two progeny, it's not what they call expressed. So they're carrying it, but you don't physically see it. So these foals will survive. If we cross them, in other words, if we cross two carriers, now look what can happen. In that first cross, down at the bottom, we've got X and B. So we've got a female progeny that again is a carrier. But this is the worrying one. In this cross, the colt foal is carrying two copies of X. So it will be expressed, and it's unlikely that foal is going to live much longer. It will probably reach full term in uterus, but in terms of actually its survival after the first 3 or 4 weeks of life, the outlook is really dim. And I know now that we can test for that X gene, and we can prevent mating carriers, and that's the strategy going forward. Because if you just say, we're going to exclude all carriers from our breeding program forevermore, you might be throwing X out, but what else is going out with the bioforce? That's the problem when we got these rare breeds in the first place. Didn't expect to come here and look at cattle, did you? Anyone recognise those? Chillingham cattle. Have you heard of them? Right, they are probably our most inbred cattle breed. There's a feral, basically they're a feral population living up at Chillingham Castle in Northumberland, and they have been managed with minimum human intervention. You know, when the vet goes in here, they're going in with a dart gun. Calves, if they die at birth, you know, they lose calves behind the hedge, and they're certainly not selecting what animals a bull is breeding with. Right? There is minimum human intervention. But this population survives, and it is mega inbred. Why? Well, the reason is that as well as accumulating deleterious alleles, we also purge them through natural process. The problem is you cannot predict the rate of purging. And in the case of Chillingham cattle, they know that actually the rate of purging exceeds the rate of accumulation. So providing they're left to their own devices, completely random mating with minimum human intervention, they will survive. But if you DNA tested them, they all come out virtually the same. They're almost clones of each other. So not all inbreeding is bad, but for the vast majority of us in the sphere of animal breeding that we're working with, we need to manage it. I know we've got Exmoor ponies, for example, represented here, and within the whole stud book of the Exmoor pony, I know full well there are subherds that are highly inbred, but that are managed in a very special way, and they are viable going forward. And part of the reason for that is because of the rate of purging within that subherd is exceeding the rate of accumulation. So it's not impossible that our breeds can survive if they get more inbred, but it's not the norm, okay? My interest was originally Cleveland Bay horses, and at a time when home computers were coming on stream, becoming much more common in the 1990s, and I researched around, and actually the world of captive wildlife management has been a really good friend to livestock breeding, because Walt Disney, who made a lot of money out of fluffy animals, pumped money back into captive wildlife breeding and into research. And the American Zoological Association, Chicago Zoo, Lincoln Park Zoo, they developed tools by which we can assess inbreeding and also manage our breeds going forwards. And if we look at the rate of increase in inbreeding in Cleveland Bays, I've only graphed it here from the 1900s. You can see it was very low back at the turn of the century, the 1900s. It goes up, and then I better explain away a sudden dip, because everything says that shouldn't happen in a close population. What happened was the two stud books got amalgamated. The old Yorkshire coach horse stud book was becoming so small and unviable that the two societies agreed to combine the stud book. So there was effectively an influx of outside genetics. That's why it decreased. But if you go from 1930 onwards, you can see that inbreeding just increased and increased and increased and increased. So that by the time you get to the turn of the millennium, we've got inbreeding in excess of 20%. Let's put some figures on it. We know it's not a good idea to marry our cousins, okay? A full cousin mating is 12.5% inbred. Equally, brothers and sisters is even more bad news because that's 25% inbred. So Cleveland Bay's at 20%, way past first cousin and well on the way to full sibling mating. That's where it's at. Very, very close gene pool. And that was, how the hell do you deal with that? Do you just go for looking at the whole pedigree and try and work out what's least related? Where do you go? That's what we were dealing with. Inbreeding that's not just high, but it's increasing at an astronomical rate. Just to put it in context for other breeds, because I know we're not all, there are plenty of Cleveland Bay breeders here, but there are other breeders as well, and I'm involved with a lot of other breeds now. Dell's pony. Yeah. But the scale on these graphs is not all the same, okay? You need to just bear that in mind. But you can see the gradients of the graph. So for Dell's ponies, it's increasing. For Exmoor ponies, it's increasing, has plateaued off, but it is increasing. Highland ponies, probably the most recent breed that I've come on board looking at. Again, we've got inbreeding in purple, increasing and continuing to increase. Inbreeding, how related I am to myself through my mum and dad. And if you breed dogs, you'll be aware the Kennel Club has got an inbreeding management tool online now, all super duper high flying. It's only half the story. Because if you just look at minimum co-ancestry, minimum inbreeding matings, you might think you're solving things in the short term by producing animals that are not highly inbred, but you leave yourself nowhere to go in the future, because what you actually do is you mix up all the common and rare alleles. It increases the frequency of the common ones and you lose the rare ones. So it's not a long-term strategy. What you need to do is think about not just how related an animal is to itself, but how related it is to other animals in the population. So kinship is how related I am to you, and to you, and to you. And I've got an individual kinship with everybody in this room through some common ancestor way back, whether that was William the Conqueror, Hubbard the Dane, or whatever. Okay? Kinship. It quantifies the relationship between two individuals. And again, it's got a definition. We can quantify it. It's the probability that two alleles randomly chosen at a neutral locus. There'll be a test on this later. I want you to remember it. You won't be allowed to leave until you can. Identical by descent. So full siblings are 50% related. Parents and their offspring are 50% related. Half siblings, 25%. Okay? In simple terms, if you start off with unrelated parents, actually kinship coefficient is normally twice the inbreeding coefficient. Let's have a quick look back at our chart again. Up at the top, we've got two completely unrelated parents. F, breeding coefficient is 0, and there's no kinship, they're not related. But if we cross them, the cross that we did before, in the second generation, we haven't got any inbred progeny, but the progeny are related 50%, 0.5, the kinship coefficient, because they share the same parents. If we made them, now we're inbreeding coefficient of 25%, we had that before, and the animals are actually 75% related because of the compounding of the relatedness. And if we carry on, they double as we go on down. Inbreeding, how related I am to myself. Kinship, how I am related to Lucy or to Tullis or to any one individual, other individual sat in this room. Mean kinship is how related we are on average together, whether that's us sitting in the room, whether it's your stud book, whether it's all the horses in the world, we could work out a mean kinship. Okay? So it's the average of all the related coefficients of all the animals that we are working with, and that's usually all the living animals in our stud book. I say living because those are the animals that can continue to contribute genetically. Obviously, with modern technology, we now need to include animals that are deceased but have got genetic products frozen that could, in theory, contribute to future generations. And to be correct, we do include those in calculations of kinship and mean kinship. So it quantifies the relationship of an individual to the rest of the population. It's a really useful tool to see how genetically healthy a population is. Okay? Low mean kinship generally means a population is fairly healthy because it's got quite a lot of diversity. It's also a good way of identifying genetically important animals in a population, because the lower kinship animals, the lower mean kinship animals, are most likely to be the one that are carrying the rare genetics. So if you're working with restricted funds, but you want to do some stallion collections, for example, and prioritise stallions for collection, the ones that are carrying the genetics at greatest risk of being lost are the ones that are of lower mean kinship. So it's a really useful tool for a variety of ways, not only for looking at matings in the future, but for cryopreservation as well, and prioritising animals for collection. Now we come to a concept that some people find a little bit hard to get their heads around. How big's your population? Are we actually just counting hooves on the ground or number of noses looking over a fence? No, we're not. Because ideal populations are a bit like the Chilean cattle. They're infinitely large, you've got a huge herd. They undergo random mating with little human interference, so there's no human choice in what's being bred and contributing to the next generation. But equally, every animal, whether it's male or female, gets an equal chance to contribute to the next generation. This never happens. Generations don't overlap, they sort themselves out very nicely. But real populations, well, we can know ourselves what real populations look like. We can't afford to keep an infinitely large stud book with restricted size. We've got unequal sex ratios. That's a real, that's horse breeding. We do not keep as many stallions entire as we do mares to breed from. It's just the nature of livestock breeding. We've got unequal family sizes. There is human selection. As a horseman, I look and I go, there's no way I'm going to breed from that. But equally, that horse tracks up properly, it meets breed standard, and I actually think that would be a really good stallion to cover my men. And generations overlap. If you try to unravel a whole stud book, it is really hard to separate it out into coherent generations. So to deal with that, what geneticists and population biologists come up with is the idea of effective population size. And for example, we might have a field out there with 1,000 horses in it. If we took all of Edward's recipient mare population and stuck them all in the field and counted it, you might have, let's put round figures, 1,000 mares. But how much genetic diversity have you actually got? If you pulled all the genetics out and you thought of it as Lego bricks, and then you sent some 3-year-old kids in there and said, build me as many individual horses as you can, they wouldn't be able to build 1,000 unique horses. So effective population size is a bit like how many unique horses can you build from that pile of genetics. And it is, from a population biologist point of view, the gold standard way of measuring population genetics and measuring the health of a population. It translates to the size of an idealized population that is losing genetic diversity at the rate of a real population. So it's a good measure of how much genetic diversity you've got and how much you're losing. And it can be measured in a number of different ways. The most common being from the number of breeding males and females. And if you think back to how RBST used to report to DEFRA on the state of the nation of our breeds. It used to report based on a multiplier and the number of breeding females. Now it's changed to the number of breeding males and females. But actually, both of those are incorrect. They've become accepted within scientific circles because they've kept appearing in the literature. But the one real way to measure effective population size is to calculate it from the rate of increase in inbreeding, which is what we looked at in those graphs earlier. And anything else is wrong. Okay, so if you go onto DEFRA's website now and look at the effective population size of your population, I can tell you, for example, for Cleveland Bays, the picture is completely different than DEFRA is reporting. In terms of population biology, there is a good rule of thumb that's accepted by the United Nations, the Food and Agriculture Organization, which is what DEFRA report to, so further up the tree than DEFRA. And that's what they call the 5,500 rule. And that is that if you've got a population that's got the effective population size measured through inbreeding of 50 or more, it is likely to be viable in the long term. If it's less than 50, it probably isn't viable. And equally, if you measure it through census, through the number of breeding males and females, if it's 500 or more, it's likely to survive. If it's less than that, it's really unhealthy and it's heading down that extinction vortex plug. So that's the 5,500 rule. Greater than 50 for short-term survival. And to do that, you need to avoid inbreeding. Greater than 500 for long-term survival. In other words, it's got the ability to change and to adapt as circumstances change. You need at least 50 animals in your idealized random population to get enough genetic diversity retained to be able to maintain the breed into the future. And for the long term, you need a population of at least 500 to avoid serious genetic drift. There's the reference if you want to go back and Google Franklin 1980, but that's accepted by the Food and Agriculture Organization of the United Nations. Okay, so knowing all that, how the heck do we manage our pedigree populations? There's a number of strategies. One of them is you can divide them up. Keep the population, the subpopulations completely isolated. So in other words, if you took all the fell ponies in the UK and you sent a third of them off to Australia, assuming there's none out there, and a third to North America, and leave a third here, and then you let them breed and manage each of those subpopulations in isolation, they will generate diversity. They will slowly become differentiated from each other. Even though we started off with the same genetics, through mutation, they will become different. And that's really healthy. But the problem is, there must be absolutely no exchange between populations. So you're not going to collect something out in Australia and ship a stallion back or ship frozen semen back. It just doesn't happen anymore. Where's the problem? It's economically not viable because of the general nature of breeders. You will get genetic exchange between populations. So that, it throws a real spanner in the works of that one. Random mating with sync and shilling and cattle, that could work, but we don't, we call the vet when things are going wrong. We make stallion selections. So as horse breeders, random mating is not an option. Or we can manage inbreeding. And that, through the captive wildlife population, has been proven to be a viable strategy going forward, albeit that in the zoo community, we've got a zookeeper with a zooman's hat on who can actually dictate to breeders or to zoos around the world what breeding program is going to be. They can decide what orangutans and gorillas are going to be shipped from North America to Japan because it's genetically important to do that. DEFRA and the EU Zootech regulations actually enshrine in law your right as a breeder to do what you want. So we need somehow to provide a tool that can enable breeders making individual choices to actually make choices that are good for the whole. And minimising mean kinship is a really viable strategy. There is one better strategy, that's called optimum genetic contributions.
But it's been shown that where you've got compromised breeds with not a lot of genetic diversity in the first place, optimum genetic contributions looks at not just the genetics, but it also looks at traits. So, for example, Icelandic horses, I keep calling them ponies and getting in trouble. Icelandic ponies have a really good scoring system that looks at the genetics and gait and scores. And you can do optimum genetic contributions when you've got the luxury of a lot of diversity. I know that with Irish drafts, they use a system called linear profiling that looks at the stand of the horse, the stamp of the horse, the makeup of the horse, looks at the phenotype and scores for that. Well, if you have the luxury of the diversity of Irish drafts, you can do that. You could run optimum genetic contributions, but for our rare breeds, it would actually be damaging because you would be throwing far too much away with the bath water. But mean kinship, it will reduce the rate of increase in breeding. It will prevent us crossing those red and blue Smarties. We don't want to dilute the blue Smarties with loads and loads of red ones. It maintains as much genetic diversity as we can. You can never stop genetic drift completely, but if you imagine genetic drift as being a pouring tap, we can screw the tap down and just get it dripping and equally control the rate of drip and get it down to almost zero. And it does help us, as I say, identify the genetically important breeders, the ones that we want to be, as societies, for example, encouraging people that are owning these low mean kinship mares that have never been bred from, that are still viable genetically, get them bred from. Or if we've got colts that are coming up to being cut, then perhaps we can use some of these modern technologies. If they're very low mean kinship colts, perhaps we can, if we can run them on to that, find a facility to run them on until I think at least 28 months and preferably older, but get them old enough so that we can do epidemic semen collection, get testes here and play the magic here. We can at least make sure their genetics are still around to pass on to future generations. We talked a little bit about DNA, but what about genomics? Because everything that I've talked about, measuring inbreeding and measuring mean kinship, it's a probability because it's taking the studbook data, it's looking at pedigree, and it's doing probability, where those genetics came from. Genomics would actually tell us what genetics our horses are carrying, not a probability, a fact. We've already looked at parentage verification using 16 markers, but we know that the genome of the horse is much, much bigger than that. Complete genome of the horse, 2.3 billion genes, as I said. Nowadays, there is an increasing move to study not all 2.3 billion because of the expense of doing it. It can be done, and it has been done. But to look at a profile of different markers, so SNP, SNPs, single nucleotide polymorphisms, big mouthful. But instead of looking at just 16 markers from the microsatellites that we're doing at the moment, this is looking at around about 50,000 or even 680,000 markers, a much, much bigger panel of markers. And there are moves away from using microsatellites for the International Society of Animal Genetics to get a much bigger panel of markers for parentage. We will one day routinely use a SNPs panel, and we'll have much more genetic information. We have information about genuine inbreeding from what we call runs of homozygosity, expanding on what we looked at in those parentage verifications before. As a breed society, pedigree is really, really useful, but it depends how robust your pedigree is. There's big research that shows that if you've got a deep stud book, Cleveland Bays, for example, going back at least 36 generations, thoroughbred stud book going back even more, Suffolk stud book going back plenty. There's a really good correlation between breeding measured from pedigree methods to that measured by genomic methods. That's great because it means if you have got a deep stud book, you don't have to fully genetically test everything simply to work out your breeding program going forwards. There's ways of doing it. You can work out your mean kinships. But on the other hand, if your pedigree is shallow, there's a greater degree of uncertainty. And I'm involved with Nottingham Trent University on a genotyping project involved with the Aeroscape Pony Society, and part of the reason for that is we are running a breeding program, a breed advisory scheme that I'm going to look at in a moment, that includes the Aeroscape Pony, but of all of the breeds, it's the one with the shallowest pedigree. So it'd be nice to know that there is a good correlation, or if there isn't, to be able to transfer across to molecular mean kinships. Okay, as breeders, we think about different things. We wear different hats. I can remember what I used to do back in the 1990s, breeding Cleveland birds. First of all, you want to look at what you're breeding from. You know, if you're a mare owner, you will only breed from a mare that's worth breeding from. You will look to a stallion that you like for a whole host of reasons. You know, does it meet breed standard? Does it track up? Or is it dishing so much it's like overseas chariot, it's going to wipe out everything along the way? Okay, you will only breed from animals that are appropriate. All of those things, using our breeder's eye, is measuring what we call phenotype, the makeup of the stamp of the horse. That includes temperament. And then we're looking at genotype. You know, how does the pedigree work? And I, like a lot of you, who have got the stud books off the shelf and you've been flicking around through and drawing up pedigrees. 5-generation pedigree is no problem drawing that up, even before the times of computers. 8-generation pedigree, just a minute. How much sellotape have we got? We start sticking together bits of A4 paper. And we're now over the kitchen table. If you want to go to 10 generations, that's an awful lot of paper, and you're probably off the kitchen table and onto the kitchen floor. And it just keeps multiplying. It's the Chinese chessboard thing. And that's how I realised that actually these horses that have got a really deep pedigree, we need to do something different. We need somehow to be able to analyse the pedigrees and do all the things that we would do by eye if only we could deal with the numbers. If you actually look at the real numbers behind that exercise, at a 5-generation pedigree on that right-hand side, there are 32 animals. No problem, we can work that out. If we go to 10 generations, there's just over 1,000, and that's why you're on the kitchen floor and you need to go down the shops and get another reel of sellotape. At 20 generations, your kitchen's not big enough and neither is your garden. You've gone to Wembley Stadium because there are over 1 million animals in that 20th generation. But hang on a minute, Cleveland Bays, at that time, we could do 32 generations and we can do another 4 or 5 now. Well, at 30 generations, the number is enormous. It's not in millions, we've now passed 1 billion. So to deal with a 30 generation pedigree is just humanly impossible. And that's why to work out and breeding from animals that are really deep pedigree, to look at common founders, we have to resort to computers. But it's only doing an expansion of what you do when you get your stud books off the shelf and you look at pedigree. It's not doing any witchcraft, it's looking at that pedigree. Just out of interest, anybody tell me how many horses there are in the world? Anyone got a stab at it? Current world population of horses is estimated at 58 million. Hang on a minute. At 20 generations, we're at 1 million animals. At 30 generations, we're at 1 billion. There aren't enough horses in the world. They've got to be at 30 generations. Somehow they've got to be related to each other because there physically are not enough horses in the world today. There is relatedness, and that's why our animals, even before our stud book, they will have been related. The simplistic way of looking at pedigree was to look at bloodlines. You know, we all do that, don't we? We get the pedigree out off the shelf, and then we look at sire lines of the sire of the sire of the sire, and we look at the dam of the dam of the dam. That's relatively easy, and you can go back a lot of generations, and you divide your stud book up into female families or whatever. The problem is that the further back you go, the less animals of that pyramid you're taking into account. By the time you've gone back 5 generations and you're only looking at male or female tail lines, you're ignoring all of those genetics. And that's contributing to genetic diversity. So it's really important. Male and female ancestry lines are important because females carry what they call mitochondrial DNA that's unique to that female ancestry line. And it's only passed on inherited from the dam. Equally, we now know that on the male side, Y chromosome DNA is only inherited down the patchy line. So they are important, but we have to be careful we don't ignore all those genetics at the centre of the triangle. And if you can imagine, as we expand that triangle back past 10 or 20 generations, it's a huge amount of information. So, some of you will be familiar with this. Adopting science from the zoo community, using mean kinship, what we've come up with is adopted a system called SPARKS. That used to stand for Small Population Animal Record Keeping System. And that's where the name came from the zoo community. And we can look at how inbred a foal would be if we made two particular animals, but equally, we can look at the kinship of those animals and make sure that we aren't mating either very closely related animals or very distantly related animals, so that we have maximum choice going forward, not just the generation that we're breeding now, but for generations in the future. And to look at it in a little bit more detail, it's grown over the years and become simplified because it breaks matings down into 4 categories, following traffic lights. And the matings that we want to encourage, they're matings of animals of similar mean kinship. Now that might sound, hang on a minute, you're mating animals that are going to become inbred progeny. But because we can measure the inbreeding coefficient, we can make sure they're not. So in other words, we're not bringing together the red and the blue Smarties. We're mating red Smarties with red Smarties effectively, but they just happen to be red Smarties that aren't related to each other. We want to make sure that the inbreeding coefficient of the progeny is lower than the average for the breed anyway, if we can. And all breeds are different. We set different thresholds for the different types of spark sheets for different breeds. But we make sure that we're trying to make sure that inbreeding doesn't increase year on year on year. So we're controlling that average inbreeding within the breed. But not everybody can do that for various reasons, whether it's geography, whether it's simply family history. You know, my father would never go to that stallion owner, and I'm not pushing his threshold. So there's all sorts of different reasons. Best of the rest matings, perhaps not as ideal as the green matings, but we've still got animals of similar mean kinship. That's really important because we don't want to lose the blue Smarties. We don't want to lose them through genetic drift by mating animals of very different mean kinship. We want to make animals, these are going to be slightly more inbred than the ones from green matings, but not so high as it's going to screw the average. And they're the ones that we need to try and encourage people to do if there's no green mating, either for geographic reasons or simply for some animals there may be no green matings. Now we're starting to get through to the matings that actually aren't really good population-wise. These are mating animals of very dissimilar mean kinship. We're going to lose the blue Smarties if people continue to do these orange matings. We're also going to get higher inbreeding coefficients. That inbreeding coefficient might still be less than the threshold because the threshold is set and it's going to turn things red. But yeah, they're the ones that where we're going to lose the rarer genetics by swamping them with the more common genetics. And these are the ones we want to avoid because these are the ones that are producing highly inbred coefficient animals, no matter what the mean kinship of the mating is. So we've got animals of very different mean kinship, but equally very high inbreeding coefficient. Different thresholds, I've put some examples up there. For Cleveland Bays, we set the threshold at 0.2 for 24%. Bear in mind that full SID mating is 25%, but we've got an average inbreeding of 22% at the moment. Eriskays is quite a lot lower, 18%, Suffolk's lower again, and some of the other breeds we managed to pitch in at what would effectively be full cousin mating. So those progeny will be highly inbred, and if they in turn went on to produce highly inbred progeny, we've got greater chance of accumulating homozygosity, greater chance of passing on those deleterious alleles. So they are potentially highly damaging for the breed as a whole. We started doing this with the Cleveland Bays way back in 2000, well, before 2004 unofficially, but officially since 2004, when it adopted the scheme. Cleveland Bay Style Book is managed globally, so the information is circulated globally. It perhaps was adopted in the USA more than in the UK to start with. But over the years, if you have a look at what was happening, I showed you that graph before, up to the turn of the millennium in 2004, now look what's happened since. It's the same graph, but I didn't point it out. We've actually managed to plateau out in breeding. It isn't increasing at that horrendous rate, that it was before. We've managed to stem the increase in breeding, and that's over best part of 20 years now. So that's about 2 generations of livestock breeding. That's not a prediction. That's a real world example of what can be done when the information is out there and breeders work together. And what's that done to effective population size? Well, measured way back in 2000, before we introduced the scheme, effective population size for Cleveland Bays was right down in the dumps. And I talked about 50 being the minimum for a viable population. Oh, it was worse than that. We were down 19 in 2000. And it's bumbled along, but it's slowly increased. It took a long time. It took till 2015, at least 10 years, one generation on from when we started the scheme, but it went through the 50. Now it's away. Look what's happening. It's increasing. Doesn't mean we're getting more foals on the ground. But what it does mean is we turn the tap off. The genetic diversity that was retained in the breed at the start is still there, and it's there for future generations as well. You know, for long-term sustainability of the breed, we've got to manage phenotype and genotype. We've got to adopt information that can help us manage inbreeding. This information on its own is not the be-all and the end-all. It won't put more foals on the ground. You know, there's only dedicated breeders with really big pockets that will do that. But it's another tool in the breeder's toolbox. So, having got a scheme in place, we need to monitor inbreeding going forwards. We need to look at mean kinship and make sure it's under control. It will never be zero, the rate of increase in inbreeding, unless you open the gate to the outside and bring in outside genetics. Trying to do it too quickly, we know from modelling, you actually get a rebound and you've got nowhere to go in the future. It keeps the maximum reduction that you can get. So it's important to do it steadily and slowly, using that idea of managing inbreeding through controlling mean kinship. And that's why we do it that way, not directly. So things to consider when you're looking at matings: breed from as many mares and males and females as possible. Premium license stallion schemes are sometimes counterintuitive because it concentrates breeders' minds on a small number of stallions. And I know the Shire Horse Society have, in the last 2 years, shifted away from their premium stallion scheme. It's coming to an end now, so that they encourage breeding from a wider base of stallions. That's going to maximise effective population size. We need to make sure we don't overuse any one group of stallions. So, for example, and I know he won't mind mentioning it, but Bruce at Toolkit Stud brought Craig Hall Jensen here, the Suffolk stallion. He really well marketed him and got him high profile. A lot of Suffolk breeders used him, and in the next year, 11 out of a relatively small crop, year's foal crop of Suffolk horses were by Jensen. And yes, it preserves his genetics, but it's actually not mega healthy to have a foal population skewed to a small number of stallions. So we need to make sure we try and broaden the stallion base and encourage breeders to use as many as possible by presenting as much information as possible. In the future, native equine breeds, yeah, it's a tool in the toolbox. I'm now doing SPARKS for Cleveland's. Eriskay's with a second on board, Suffolk's well on board now, and we're Shire Horses doing some work, and we've got out there for Exmoors. Certainly, Dales have been using it for a couple of years. Highland ponies, hopefully we're very close to being able to issue the SPARKS sheets now. Just need to finalise your stallion list for this year, and they'll be out in the near future. But it is another tool in the breeder's toolbox. Leaves you as breeders in control, and societies can take an overview of what's going on, but they can't dictate what you do. It does avoid those highly damaging matings. And if you don't get stacks and stacks of green matings, at least if you can turn off the red matings, you're doing a lot of good. It maximises the amount of diversity that isn't going down the plug hole. And we need to try to encourage the owners of low-banded mares to breed to appropriate and preferred stallions. Quite important, green matings for low-banded mares. The science for this is proven, it is peer-reviewed and open source. If you go and Google ecology and evolution, 16 years of breed management, Cleveland-based, you'll hit this paper that I co-authored with Philip Wilson, who used to be at Rare Breed Survival Trust and is now at Nottingham Trent, or certainly is at the moment. And you can access that, and it goes into a full review of what we've done and explains the science behind it. So that's in ecology and evolution. You don't need to write a link down, you actually just hit it, it's high on the Google hits now. And breeding for the future of our native breeds, what is clear is that uncoordinated breeding doesn't work because we accumulate inbreeding too fast. Look, we need to direct our thoughts at managing effective population size and monitoring it, maybe as breed societies, but more importantly, Rare Breed Survival Trust and DEFRA are monitoring it, definitely. That year-on-year, certainly generation-on-generation analysis is really important. And, as an umbrella organisation, Rare Breed Survival Trust takes that on for DEFRA. And maybe if your breed isn't on board with SPARKS at the moment, and there's still some of the high priority breeds that aren't, then through Rare Breed Survival Trust, you can get access to the self and that piece of software, and it's specific. As long as you've got a digital stud book, we can work with it. It isn't commercial software yet. We're working with a company called Cloudlines to get it there. They're not quite there yet. Part of the problem is that it isn't desperately commercial for rare breeds. But I've just done a piece of work for thoroughbreds that might lead to SPARKS for thoroughbreds, and then suddenly it is commercial, and the rest of the software development could be done. So, hope is not lost. If I go under a bus, we don't lose all of this. And as I say, the EU ZTEC regulations protect you as a breeder and make sure you can do what you want to do. But if you know you're making breeding choices that are appropriate for the breed as a whole, that's great. If you want more general stuff on population management of livestock, go on this website, breedingfordiversity.com. This guy's interested in canine breeding, but the science is all there, and it's well reported. A researcher in the Netherlands called Pierre Ongeluk. So it's exciting times. SPARKS are flying everywhere. Sometimes there's now as clear as breed societies in their way to use the information, but those are the breeds that I'm working with at the moment, and hopefully very soon we'll be able to add the Highland ponies to that. So thanks very much.